All right. So welcome back, everybody. Well, aside from those two of you that were here last week. And welcome to a couple of newcomers I invited to the class. We've got Caleb back there, my other grad student, and Samuel visiting all the way from France. Be here for about an 8 to 10 week internship. So if you've been to France, say hello, or if you just are curious, want to know anything more, go bother Samuel. But actually, it probably would be pretty nice if anybody was interested in sharing kind of where to do things around here, things like that. I mean, I've given him way too much information on research already this morning, so I'm sure he could use some other information other than that. Okay, so how did the senior design competitions go? I heard some good things. Enjoyed it. I see a nice T-shirt. Trophies, good times, no major failures, anything like that? Awesome. Nice. Good. All set and ready to wrap things up with a bit of writing and for my class. Gotta write my paper, right? Okay, well, yeah, and the deadline, by the way, is Friday. When I went and I thought I had corrected the deadlines, and then I went back and I was like, oh, well, I had got it in the rubric correct and uploaded. I think I uploaded that one. I hope I did. And then it was different in all the different spots aside from that. So I was sorry about that. But Friday would be fine for the next deadline. I'll try to get you feedback within a week or two after that, and then that gives you a week or two for finalizing and submitting a final paper. So if you didn't yet, I encourage you to go take a look at the lecture from last week. I will post those notes this week, so you'll have those also. A lot of that was writing tips, but it was also calling everybody out on plagiarism. So recognize that you were probably plagiarizing, and I explained what I meant and why that's all right at this stage. So, not submitting any issues with that or whatever, but just the proper, plagiarism does not have to be intentional. Right? It could be accidental. So what I was referring to is if you didn't cite where you got the information adequately, properly, then the assumption is that you were the one who went and found out that the opinion of Nicaraguans is XYZ. But you didn't do that, probably. And if you did, maybe you could even say, oh, citing such interviews or whatever, right? Or mention on your trip you learned from, So just wanted to say, not thinking anybody has done something wrongly, aside, as a draft here. Some of you didn't even have citations yet. I understand. I told you the draft that just wanted to see kind of your thesis statement. So I was trying to point you hey, look, and the scores, by the way, are intentionally pretty low on that. So don't get too freaked out about that. But just to show you, if you carried forward that effort, this is kind of the expectation of where it would end up, but I know that's not your intention for the most part. Some of you actually put a lot of effort in and was okay, I was still trying to be critical to say, here's some pieces that I think need a lot of improvement. So for your paper in that regard, just wanted to comment on that. All right, so today, well, you know what? Let me bring up the syllabus real quick to kind of touch base with you where we are. I need to formalize for you our plans for group presentations. I'll talk to you about that on Thursday, because I have not formalized that yet. But just a quick note on where we are and kind of what I expect for the rest of the semester.
Are the grades from the introduction paragraph going to be included in the total grade of the paper? It was out of 25 and the paper's out of 100.
Okay, yeah, so let me explain that. So in the rubric, we'll do that first. In the rubric, basically I had just sectioned off each category as here's 5 categories. And I wanted to just simply rate them on a 1 to 5 scale, approximately. Then I'm doing 0.5, so I guess I should have done 1 to 10. But anyway, just as ease of oh, this is about 3 out of 5 in terms of how good, let's say, your grammar and whatever is, based on my judgment there. That's just a snapshot. When I do a final calculation for these papers, what I'm going to do is take the entire final product and say, and check it against essentially this rubric here. Okay, so your final grade for your paper is primarily going to be that final submission. Okay, if, like, let's say, I'm trying to think of, maybe I would put 5 to 10% of the grades contribution from the draft here, or if it was like really clear to me that there was no effort in either of these, and then suddenly you have something that looked like ChatGPT wrote it, maybe I'm very concerned and am going to act accordingly. But chances are good what I'm doing is giving you harsh feedback here and here as best as I can, so that this one is great and gets a great grade. Okay, so that's my hope for you, and I'm not at all going to average these three and you get the final grades at average. If anything, it would be more like this is going to get 90% of the weight, and maybe I would do something. If I wrote something specific here that's been in the instructions, go by that, because that's what I need to go by. But that's my plan right now, is approximately what I just said, unless this tells me otherwise. Okay, so but for sure, I'm not going to say like it's a third of your grade is this draft that you just did, right? Okay, so where we are now, done with senior design and all that. Today's date, the 16th, right? Yeah. Okay, so today I'm really going to actually talk about this topic, and we'll probably save this topic for Thursday. It gives us space still in the next week, perhaps, to talk about this guy. And I don't think, for this class we have what, 3 groups? We can actually probably do all the presentations in one class. So I'll have kind of a practice session in preparation for the exam, like let's work a few problems together in class, kind of a thing, getting a scope for what I'm expecting, and we'll give you time so that you can do presentations one or both of these days, and a few more lectures that are more topical, kind of research interest type stuff. Okay, so if you do have anything you really wanted to hear about, if you're sick of toilets, you don't want to hear anything more about toilets, yeah, maybe I'll find something different, or that kind of thing. Just let me know if you have some feedback about that. Thursday, I will come to you with some specifics, like, okay, here's and an update to this to say, here's where we are. We're going to present on this day. Get ready, all that. Okay. All right. Okay, so today I want to talk about solar disinfection. So solar water disinfection, some challenges to using sunlight. There's the common moniker or phrase that sunlight is the best disinfectant. And I like to say that's only really true if you're in outer space, because thankfully our ozone layer and oxygen in our atmosphere absorbs so much of the UV that by the time it gets to us, if we have a long exposure, maybe it'll give us a burn, but it's not so detrimental, right? And so maybe in the space station, sunlight might be the best disinfectant, right? Okay, so can we use it though, and how would we use it if we can use it? To what extent can we use it? Well, it turns out it is possible to directly use sunlight. There's a few mechanisms of action that are going to happen, and so I'm going to dive in a little bit to each one of those, and then talk about what else can we do with sunlight to enhance this, to catalyze some disinfection type reactions or other water purification, and what all would that look like? So the basic principle of SODIS, solar disinfection, this is the common abbreviation. To use SODIS, we would take a PET bottle, that's polyethylene terephthalate. Those are Coke bottles, clear plastic. Turns out different plastics will absorb different amounts of light. If you've got a color to your bottle, that's going to have some dye coloring stuff, and it's going to absorb some of that light, and it's not going to be so useful. So we take clear plastic bottles. These actually transmit UV pretty well. Talk a little more about what types of UV and all that in a few minutes. We've already touched on it before, but these ones will allow the UV to come through, go into the bottle, and it will also provide some heating. So we'll get some temperature effects that'll help speed up decay of pathogens and reactions that might kill them and stress them out. And overall, if we leave them under intense sunlight for 6 to 8 hours during the day like this, they'll be a little cleaner than they were before. Okay, now you notice I said a little cleaner. That's a good thing, but that's also pretty challenging to think to yourself, oh, I want something to drink, let me clean this water. First of all, we kind of take it for granted. Oh, I need to clean it, right? Can't just grab it from the water fountain. Okay, so now I'm gonna set it on the hot rooftop for 8 hours, and then I can have my water, and it's gonna be hot and not so pleasant. So there's a few things there to keep in mind, that this is not necessarily an ideal solution, even if it's maybe helpful. Okay, so the basic principle, we're after disinfection. We're really not expecting much other treatment. So if you have some toxic things, anything like that, we're really not expecting much progress benefit from there. It's actually only a small fraction of the solar energy that's going to do that direct killing. Specifically, we call it UVB irradiation. Some of the UVA, it's a spectrum, so some of the photons, the UVA can also do it. This is kind of like the stuff that gives you a bad sunburn versus the stuff that's triggering your body to do a suntan. The photons in that region can kind of do both, but the ones that are higher energy are doing more of the damage, and the ones that are lower energy are still triggering your body to produce more pigment, and tend to do a little less damage, and the pigment's actually helping prevent that as well. And part of the reason those actually work, whoa, okay, hello. Part of the reason this light works is what we would call a photosensitized reaction, where we have some molecule absorbs that light and then gets kind of excited, and then it can either directly react with something else, or maybe it produces what we call a reactive oxygen species. Then that thing goes and reacts with something. Maybe it's DNA, maybe it's some proteins. It's just going to cause some random oxidation events. If you think about a burn, like let's say a physical burn, it's hot, it's causing things essentially to oxidize, right? Or to be otherwise stressed out from that heat. That's a very similar process to if we're causing a bunch of oxidation reactions inside your skin. So your skin as an organ is now getting damaged by this oxidative stress kind of stuff. So that's what we're intending to use with the microorganism. So you can imagine if a bacteria cell has some organics inside of it, even the DNA, maybe the DNA molecules itself gets excited, or something that's neighboring it, and then you have some reaction accordingly, and it's stressing the organism out. So I'm talking about the solar spectrum here, and this is actually some data we collected here several years ago using a fun toy during the 2017 eclipse. So any of you get a chance to see the eclipse at all here? Some of you, I mean, it was here, it was like raining, so it's kind of no fun. Pretty boring, right? Well, in 2017, I think it was probably September or something, we had a solar eclipse, partial. It was maybe 60 or 80%. It was pretty neat. We took a little radiometer outside and tracked the change in how much sunlight we were getting. And so this is a picture of just the intensity of sunlight over time. So you can see we started, I think, at just after noon, and then the eclipse went by pretty quick, and that by 1:26 p.m., I guess that's maybe half of it, right? And then it would growing back. So it was over that hour, we were decreasing in light intensity. So that's just kind of neat. Can share a little bit of an eclipse with you here, in case you missed the other one. But overall, the eclipse is not changing the color of the light coming in, right? This is just literally changing the intensity. It shouldn't be doing anything else. So in addition to the fun of the eclipse, I just wanted to point out what kind of UV light we're getting. So 400 is kind of where we say there's the cutoff for the visible and the UV. So to the right, the higher wavelength, and remember, if you have something traveling on a path that's going like this, you compare that to something that's going like this, one of those looks like it's more energetic, right? And it took more energy to write it. There's higher energy in the photons with a shorter wavelength because they're kind of more active in that sense. So they're both traveling at the same speed, one's higher energy. Okay, that's the way to think about it. So the visible wavelength is kind of about 400 to 800, 400 to 700 something. So this would be the visible region. Here we're getting to infrared and so on. And below here we have UV. Now we often classify it into kind of three categories, UVA, B, and C. And we generally say that A goes down to something like 320. And then B is this small little bit here. We'll just say that. So this would be UVB here and UVA here. And then below that, which we really don't get anything from, is UVC. UVC is the stuff we use when we have a mercury lamp and we're trying to do disinfection directly. That's going to cause DNA to we say dimerize, it probably have a slide on it, really just destroys the DNA, prevents them from doing anything else. It's like adding super glue to a zipper, it can no longer function. It's stuck. So UVB, this is the kind of stuff that's really giving us the sunburns primarily. Enough UVA will do that too, again, a spectrum thing. That's also a good source for us to use to destroy our pathogens. So it turns out that if we were to go to a much higher latitude, even in the middle of the summer, the sun is still going through more atmosphere to reach the higher latitudes than it would be towards the equator, right? So at the equator is generally where you have year-long the most solar irradiation, and therefore the most direct path for the light means less of the photons are being absorbed by the oxygen and stuff in the atmosphere. So at some wavelengths or some latitudes, we have limited UV. If you climb a mountain, go to the top of a tall mountain, there's less atmosphere, you're more prone to getting sunburned. So people that have ever gone up a tall mountain to go skiing or something, and then there's all this snow, and you think, oh, it's cold. You don't even think about sunscreen necessarily, but then you're getting all this light reflecting off the snow, and it's not going through much atmosphere. You get sunburned, and maybe have issues even kind of snow blindness, they'll call it. Just stressing your eyes out from that. So it really is all about how much atmosphere it's going through. So we can't really use solar disinfection, SODIS, in certain areas, right? Most of the developing country contexts would be tend to be towards the equator, so maybe it's still an option where it might be useful, but that's kind of the technical perspective on what's going on. And you can see if we were permanently in an eclipse, maybe it wouldn't work very well, right? Just to bring that eclipse right back in here. Okay, so it talks about the UVC range. This is when if we put in a germicidal lamp, you'll notice basically our UVB range kind of cuts off right around here. So this is UVB, and then to the right would be UVA. Down here is UVC. So when we intentionally use lamps that do this germicidal stuff, we can see why it's germicidal. Is this germicidal effectiveness is essentially where DNA absorbs directly, and in such a way that it can cause these cross-link reactions. So we can see that a little bit of germicidal effectiveness into the UVB range, but most of that's in the UVC range. So mostly we're protected from the sun, from the atmosphere, because most of the radiation is not doing anything with this, capable of killing the germs. That's good for us in the sense that we're safer to go out in the sunlight. Makes it possible and all that. Not so good if we're trying to disinfect our water, so then we would maybe use, these are two types of mercury lamps, low pressure mercury or a medium pressure mercury lamp. This is a mercury vapor. This is the conventional fluorescent lamps, kinds we have here, except designed slightly differently, and no coating to prevent the UV from coming out. In fact, it's a special system to let the UV through. Okay, so that's kind of the premise of SODIS, and we can kind of see why the two mercury lamps here, they both have peaks down here in the UVC range. The medium also has a big peak in the UVB, may or may not be helpful. Probably wasted energy if all we're trying to do is kill the microbes. Okay, so there's also some other things that we might consider is temperature, the turbidity. So if there's a lot of particulate matter in the water, that's may protect the organisms. Of course, if it's cloudy or rainy, your SODIS is not really working so much, right? We can't use solar power if the sun is blocked. Already mentioned latitude. So if we take a look, and I have a few pieces from our book here and there, we can kind of take a look at what types of diseases we might expect to be killed or affected by SODIS here. This in particular is kind of a description of if we use SODIS and achieve 40 Celsius. So remember body temperature, the states here, we often use Fahrenheit, 98. Celsius body temperature is 37. So this is like having a high fever kind of a thing. So high fever is not good for us, could eventually cause problems. High fever for the bacteria, viruses and all that also can stress them out. So the very best we could, let's see, I think these numbers are wrong. And I remember last time, I need to go back and see where they're coming from. Perhaps if you, like this is saying 6 hours at 40 Celsius, I don't think we are supposed to be able to get 5 log reductions. So I'm not sure, or maybe this is the amount remaining. No, this is reduction. So these numbers, I'll tell you right now, I don't believe them at all. I don't recall where I got these. Maybe this is saying we want, no. The book itself says like, yeah, maybe we get 1 or 2 log reductions. So these numbers are clearly wrong. I'm sorry I didn't go back and catch this before coming, but we're not going to trust these for the moment. But these are things we'd be interested in knowing. How well can we get E. coli, Salmonella, Vibrio cholera, Shigella, Campylobacter, all these different things. The rotavirus, poliovirus, all of these pieces, and then the parasites as well. We hope to have some reduction, but in general, some of them are going to be more or less resistant. We're definitely not getting 5 log reductions for the bacteria. I can promise you that. Hopefully we get some reduction. Okay, so temperature effects. Part of the solar disinfection is simply that we're just elevating the temperature. And if you recall, we've talked before, and we even had that incubator here, getting bacteria to a high temperature will eventually cause them to die. Now, when we did that incubator system, we were testing actually for thermotolerant bacteria. That's a pretty good indicator of the types of bacteria that can survive in a gut, because we raised it to 44 Celsius and incubated it there, and the media that we used for those ones would show up a different color if the bacteria were stressed by it and like struggling to survive, and they'd be pink, or they'd be yellow if they're kind of happier. So we could get this differential count of which ones are thermotolerant and all that. So if we take a look, okay, how do we know how much time is required at a given temperature? we could look at the survival given some amount of time. And here we're looking at something that's pretty sturdy because this is 105 Celsius, so over the boiling point of water for this particular organism. And if you only give it 10 seconds, it'll kill off 90% of them, but there's still 10% of them that survived it, right? So we can take a look and we can learn something about how much they're surviving. This kind of goes to show also if we want to truly sterilize something, our practice is to get this to 121 Celsius and keep it there for, make sure everything's at least like 15-20 minutes. So that would be the sterilization protocol. That's going to make sure all of the proteins in the biological material have been denatured. Just literally just can't stand it. They are destroyed. Okay, we can also look at some less severe cases. Here we've got 72 Celsius. So this is bacteria in raw milk. So this would be looking at kind of pasteurization type of process. And we can see that, well, if we get it at some temperature, some of them will die off really quickly in seconds. Others may have an initial shock, but then are kind of stable for a little while. But again, this is seconds. And you know what happens if you take your pasteurization process to several hours, right? Or something like that. The idea there is the idea of pasteurization in general is you're trying not to really cook the milk or the orange juice or whatever, but you're trying to cook the bacteria. So there's going to be some change in the, there's going to be some cooking involved in general, but the hope is to make it more safe that way. Okay. So temperature is part of it. If we can elevate it even a bit and then hold it there for 6 or 8 hours, then we can kind of get a feel for, okay, that's why this is having some impact. It's going to slowly, it's sort of like the contact time, concentration times time principle, where enough of the killing agent for enough time will get us there, right? Or will get us partly there. Okay, some basic pros and cons here for using SODIS as just practically speaking. Well, we can use common, very cheap plastic bottles, right? The PET bottles. So it's almost zero cost there to just put your water in there and set it on your rooftop. It's very easy to understand, and where it's relevant, there's often good sunlight. Of course, one of the cons is it's not always sunlight, right? So if you're really just relying on this only as your source to drink, that's not great. And it's probably not a solution that I would ever think to advocate for as a final solution or anything of that sort. Perhaps a disaster relief scenario where, okay, if nothing else, you don't have any fuel to boil water or anything like that, just grab a plastic bottle, do this, hopefully that'll help a little bit. So it's really not sustainable in the sense of, is anybody actually going to do this for their normal daily source of drinking and be at all benefited by that? I kind of doubt. They actually really only inactivate about 90% of bacteria. I have some homework to go do and figure out what the crazy that table was talking about. A lot of protozoa and viruses are just sturdy against it. So protozoas are the eukaryotic organisms. They have a full cell wall, not just a cell membrane. A lot of them can kind of form a cyst and just shields up, protect themselves until they can find a good environment to grow again. Like that's the Cryptosporidium and Giardia. They'll do that. They can survive outside of water that way and things like that. They're much more sturdy. And so a little bit of heat and a bit of sunshine tend to they have more defenses against that. So a lot of them will be kind of not harmed. The viruses also, they don't have as many moving parts at all. It's just a bit of DNA and stuff. And so unless they happen to have a photoreaction or something, most of them are probably going to survive. So they're just so simple and not harmed by that level of heat. They don't have the metabolism and things like that going on, and not a cell. Of course, it takes a long time to do this. fair amount of labor involved to do this as well, and then you're going to be drinking like warm water if you just directly drink it after that, right? And if you don't drink it right then, I mean, maybe cooking with it or something, but if you don't, then you know, is it going to just get kind of recontaminated as it sits there in bacteria repair overnight or whatever? You know, there's some questions there as well. So how important is that in the scheme of things? Maybe it helped a little bit. It's also not a very aspirational. It's not a very exciting something you could be proud of, right? So one of the things that when we went to Nicaragua, one of the things I love down there is how excited people are about the water systems that they've helped put into the ground, and then they get good water and they know they were a part of it, right? That's like a huge aspirational component. Is actually a pretty good indicator, if not mechanism, of sustainability, right? You know, it's quite often that when we think or talk about sustainability, we're talking about I don't know, non-human factors, or just like we're boiling all of environmental factors down to the simple things that we can maybe measure or are easy to estimate or something. But another aspect is the human drive, the human behavior component, right? Are people going to actually want this, use this, enjoy this? And I would say this aspirational component is probably a big factor there. Okay, so there comes a question. That all doesn't sound that great. You know, lots of cons, only a few pros. What can we do to enhance it? Can we perhaps coat a bottle with some material that's going to be activated by the light and create reactive stuff that goes, you know, basically create our own chlorine? And here's where actually my graduate research was on just that. Is the hope was to use these fancy fullerene materials, which is they're essentially all carbon, just like diamond or graphite or graphene, and it's in the shape of a soccer ball. That would be a C60 fullerene. They come in other shapes too. Add a few more carbons and still have this like cage structure. They're pretty neat, and they are particularly effective as photosensitizers. So light hits them, they get excited, and then that transfers to oxygen in the water. The oxygen, then it becomes really quite aggressive and will go and react with stuff. So the theory is then you can go kill your virus, and this is a light activated pathway where your sunlight can activate a material, that material can activate oxygen, and oxygen then is acting like your bleach, like your chlorine, right? So that's the scheme, and the concept is great. It's like, oh, this would be sustainable. We've got oxygen, we've got sunlight, great. And this all can happen very quickly, as we'll see. At least for some pathogens, it's especially good for viruses. But there's a lot of challenges involved, so I'm going to kind of go a little bit deep into some of the research. I'm going to try to keep it relating back to how can we make something like this applied. Okay, but I know too much about this stuff, so pardon me if I accidentally dip it a bit too deep for a minute. So before I go too far into that, another type of material that often is photosensitive is actually dyes. So not every dye, but many dyes are photosensitizers. And actually, there's a PhD student who worked in my advisors group, so Eric Ryberg. He got to do an exciting project that I thought was a little more practical than my own, and so I was always just a little bit jealous, playfully jealous, not truly. But there was a very nice paper here. They actually took edible dyes, like food colorings. Some of them are photosensitizers, and said, hey, can we add this to water and use that as that photosensitizer? It should be food safe, right? Because it's a dye. And then that will help clean the water. You put it out in the sun. And they even found that, well, as it's doing this process, the dye is also getting photo bleached. So it's kind of the dye is destroyed. So you can even check, okay, once the color is mostly gone, then it's safe to drink. And that actually tracked pretty well with some of them. Now, there were some issues. Maybe once these dyes are broken apart, they might actually be releasing a lot of iodide. Maybe that's too much for your health. Maybe the degradation byproducts of these dyes as they degrade, maybe not so good for your health. So there's some open questions there excuse me, about really how practical is that, and is it something that's actually good or not so confident. But it's a really neat thought, and it kind of illustrates some of the challenges to enhancing solar disinfection, because this, you could do it in maybe 15, 20 minutes, right? So take 6 hours down to 20 minutes is a good deal, and you're getting much better treatment with that. So one of these types of materials, so those were photosensitizers. There's another type called photocatalysts. I mean, both of them are photoactivated. They kind of both do the same sort of a thing. But another option would be photocatalysts. You're going to get a direct electron transfer reaction happening. Light hits the material. You end up splitting, kicking an electron up to the conduction band. This is actually just like our solar cells, right? So solar power works this exact same way, where we get an electron, instead of just exciting it, it's kicking it up to where it can move away, basically. And once we do that, it'll go to the surface, and then there's a few reactions you could react. You could reduce oxygen directly, get to this what's called superoxide radical anion. That typically goes through some other reactions. It's pretty reactive itself, could act like our chlorine, or a really strong one. Ultimately, we often are aiming for this hydroxyl radical, and we'll get a bunch of that, and that's really, really reactive. And on the other side, some hydroxide in the solution, OH-, will react with the hole, or the positive charge that's left behind, and also create more hydroxyl radical. The thing is, these are so reactive that they will react with themselves, they'll react with the water, they'll react with kind of anything. So they don't live in the water very long, and they'll go away very quickly. So when we think about a contact time, we kind of have to be producing these constantly, and we have to get them in proximity to the target, to the bacteria, the virus, or whatever. So that actually turns out to be the biggest challenge for these approaches. Now, what could we use these photo-oxygenation systems for? Well, disinfection, obviously. That's what we've been talking about. Some other potentials would be making a surface that's self-disinfecting, be coat a surface, hospital rooms, or water fountains, things like that. Drinking water, obviously. Wastewater also, perhaps there's some nasty stuff in there that we want to degrade before reusing it, or something like that. So maybe we could attack pesticides, because we're getting these really highly reactive things that are going to essentially do what we do with advanced oxidation processes. In some sense, that's what a photocatalytic system is in a way. So we might be able to destroy pharmaceuticals, industrial chemicals, things like that. Even caffeine. Caffeine is actually a really good indicator of human activity. If you have caffeine in a natural body of water, you know there's some humans nearby. So you can very reliably detect caffeine where there's been human activity, and kind of have that connection there. Okay. By the way, here's a nice picture. This was from one of the schools in Nicaragua back in 2012. Okay, so one example, this is going back to these fullerene materials, the photosensitizers. We're going to take a virus that we study in my lab. It's called MS2. It's very common. It infects bacteria. So we call it a bacteriophage. So it's not going to do anything bad to us as humans, pretty nice, safe to work with. It actually infects E. coli. So the worst thing that would happen is if you were to ingest some, and if you did have E. coli somewhere growing in your stomach and being a good thing for you, like sometimes that's fine and good and everything, maybe it causes a problem for those E. coli, and I don't know, maybe that would disrupt your stomach a little bit. I don't know. Probably would never matter at all. At any rate, it's a virus, and it's actually pretty similar in size and resistance to poliovirus, or excuse me, not polio, norovirus. So it's a pretty good surrogate. It's pretty resistant to UV treatment. So it's often used in labs as a good replacement for enteric viruses, so stomach viruses. So here, we were using it, we were creating singlet oxygen. This is the one that the fullerenes create. This is going to act like our chemical disinfectant. And we were generating it with some light, either visible or UV, and with a little bit of the sensitizer, the C60. It's a fancy one, but we added C60 at different concentrations here. And we watched how effective it was. So we can see with 1 millimolar of our C60 solution, we were achieving over 5 logs reduction. So that's the 99.999%. Under light that's pretty similar to these. It wasn't like super bright. It was just a little bit of light, just kind of right under small fluorescent lamps. So just some visible light, and like within 120 seconds, that all happened. So under 2 minutes, 5 log reduction. So quite fast. We could even drop the concentration pretty low and still be observing some pretty good inactivation. Okay, so this is, that actually spawned several questions because we were kind of surprised it was so effective. And that's something that I'm going to go on a bit of a tangent with, because ultimately what we should expect, just like, let's say if we have chlorine, HOCl, times, or reacting with some amount of MS2 times time, right? That's kind of our contact time thing, right? And I know I wrote this on the next slide, but dNdt, if we're tracking the number of bacteria or viruses over time, this is going to be a first order rate constant, right? So we'll have some K for that reaction. We'll have the concentration of our disinfectant. In this case, that'll be our single oxygen or chlorine. Times the number of microbes we have in there to the first power, right? That's a first order decay reaction. This is from the Chick-Watson kinetics, where it's not just a K, it's the K times the concentration. We could call that a K star if we wanted to. We've seen this before, K star times N. Okay, so that process then means that it should not matter how much, like where we got the single oxygen, it should just matter how much we have. But the data looked like the single oxygen in this particular system was somehow special, which can't be really true. So we're working on exploring that. And so here's the data we have showing just that. So we've got on the left here some log change in our MS2. That's our virus. And this is showing three trends here. One is a dye called rose Bengal. So RB, that's rose Bengal. That's a common single oxygen photosensitizer. It dissolves in water. You actually use it in like eye drops for if you go to the eye doctor and they need to check to see if there's anything wrong. They'll put a little bit of that in your eye. That'll create a color if there was something wrong or whatever, and that they do their business. And then they tell you don't get any sunlight for a little while. Like shield your eyes, protect them. You'll be sensitive. You'll be sensitive because you have photosensitizer in your eye now. And it's going to create a bunch of single oxygen, which is not good. So do pay attention if they do that. But so it's actually pretty commonly used. So rose Bengal. Sure, there's a scientific name too, but that's what it's typically called. And then we have the red triangles here, methylene blue. Another dye. And it's possible that they produce single oxygen kind of more or less effectively. And the last one, the green one, here's our fullerene C60 stuff. Now, what you'll notice if you look closely is the rose Bengal and the methylene blue, these are both 50 millimolar concentrations or 50,000 nanomolar. But they were outcompeted by the C60 stuff at 100 nanomolar. So the difference here is quite impressive because theoretically these are all just out in solution, creating some single oxygen, probably similar amounts. So they should all be somewhere kind of similar if we were to use the same concentration. But we had to drop that fullerene stuff way, way low. So it's kind of exciting that maybe this stuff is really good. It's performing really exceptionally well. But there's a question there because if we calculate the CT value, that's the amount of concentration times time for single oxygen here. So this would be single oxygen times time should give us some value that's constant for single oxygen, for this reaction, single oxygen killing the MS2. That should just be constant regardless. It shouldn't matter where you're getting the single oxygen. And so when we see a bit of a difference here, okay, maybe there's some error there, or maybe there was some interaction that was causing a bit of a difference. I'd kind of believe that maybe there's some variation. And in fact, if you look in the literature, there's some variation in the reports. But ours, for the C60, was way down here, saying that this is like 3 orders of magnitude different. That's a bit different. It's harder to say that that's just a little variation. So that was something that we're really trying to explore because this shouldn't happen. And so one thing we believe that's the answer here is the dye molecules are just completely dissolved. But these fullerene molecules, they don't like being in water, so they clump up. And so then we end up with a surface that's like all these fullerene molecules in some sort of aggregate. And then the theory is that they're going to produce a lot of single oxygen right at the surface. And some of it will diffuse away, but a lot of it's just going to be high concentration near the surface. And the way we measured it and measured the single oxygen was really only capturing the stuff that was a little further out. And so way out here in what we would call the bulk solution, there's just not as much. And maybe we measured out here a little bit of single oxygen, but then in reality, the viruses were sticking very close to the surface. We'll just say the virus here. So the viruses actually experience a much higher concentration than we thought. So that's kind of what we are theorizing here. And this graph here is going to show us again the MS2 log reduction over time exposed to the same fullerene stuff, but this time, we can take a look. It happens really, really fast if we give it this particular condition. But then as soon as we add some organic matter, what we find is organic matter is stopping the process very quickly. This is humic acid. We're just adding even 0.1 milligram per liter is reducing it quite a bit so that then it takes 30 minutes instead of 2 minutes to get a 3 log reduction. And if we add 5 milligrams per liter, it kind of stops the thing almost entirely. So the theory here is that this organic matter, really making use of the colors here, this organic matter is actually satisfying the charge at the surface of our fullerenes, because that we have a positively charged surface that tends to attract these viruses, which are negatively charged, but it would also attract our negatively charged organic matter and crowd out the space so that then the viruses can't get there. So this is something that we are still working on finalizing some data to get it published. But that's kind of the thinking, is that as soon as we add all this organic matter stuff here, it's giving us those problems. So bringing it back a little bit to how can we make use of this stuff? If we can coat a water bottle, for example, with something that's positively charged, maybe we can get some pretty good virus removal by getting the viruses to stick there and also have some photoreaction happening. That's somewhat promising, but we do have that issue with organic matter, right? So we also need some way to deal with that or to get around that, because it clearly is one of the challenges. And one challenge in particular for all of these photocatalytic or photosensitized systems is if we are going to make use of something with a surface, we have the issue of surface fouling, right? Is it going to get gunked up and prevent the activity? We also really need that target to be in contact with the surface, right? That's kind of key to the issue, and this is why we've seen like the lifesaver straws, a membrane filter in a straw or in a bottle or things like that, that we can practically use and purchase, but we don't see any photocatalytic things that we can buy, right? There's very, very few practical applications, which is pretty disappointing for how nice it sounds to say, hey, let's use the sunlight and use the oxygen, right? Seems so applicable, seems so convenient, but unfortunately, we don't have practical applications. So that's really a goal and aspiration for this type of work. Okay, so just going to show a few more examples and a couple of thoughts about how maybe we could apply this in a way that could work. Here's a log reduction of E. coli with different amounts of TiO2 photocatalyst. So we're destroying the bacteria, creating hydroxyl radicals. And this actually highlights how ineffective it can be sometimes, because we can see even 2 grams per liter, so that's a lot. That's a lot of stuff. It looks like milk even if you have like half a gram per liter for this TiO2 stuff. It's taking 3 hours before you get a 4 log reduction. Now, E. coli is a little less sensitive than virus, but still that's way too long, and this is also under probably UVA radiation. And also we notice there's a bit of a lag phase here, that kind of just showing that the surface of the bacteria is kind of protecting the guts, and it takes some time before that can be destroyed, the membrane can be disrupted, and so it starts really attacking and destroying the organisms. So that's not a great look for photocatalysis to be used, but it's something. A lot of these criticisms were voiced a few years back by actually my lab mate Ezra Cates. He's now he works at Clemson. It's like, where are we going with all this? It doesn't really work. So that just kind of describes that type of challenge and issue that really, it's not been applied. Why not? Where are we going? Like, how can we do anything about it? So, good to recognize the reality of the situation, that it's really unfortunate or lacking the needed progress for it. So, well, why not? On a technical sense, there's a few reasons. We already talked about that surface reason, and I'll elaborate I guess I'll just give a more general description of that. Part of it's also trying to use light well. If we can only use visible light using sunlight, that's a bit of a challenge. A lot of photocatalysts aren't very effective there. So maybe there's some room for improvement there. But you have to be able to get your light to your photoactive material. So absorption of light by natural organic matter in the water might be a problem. Absorption of light by whatever you're holding your catalyst on or in, so your bottle, so I'll add to this and say whatever bottle or container, like light management in general, and maybe I'll just say catalyst support. So innovations in those sectors seem to be pretty helpful too. Big one is diffusion limitations, right? So these reactive species, we say reactive oxygen species, ROS, they don't last very long. So if we did have a glass of water and we coated the surface with some photocatalyst, well, only water that's at the surface is getting that treatment. So maybe that would be good for preventing biofilms. Maybe that's beneficial still. But if we wanted to treat all of the water, we have to mix it and then mix it for some amount of time. And if we then want to scale up to do something that's a flow-through system, and we still just have that in place, even if we are mixing, there's going to be water that never spends much time with those radicals at the edges, right? Because it's contact times time. So the concentration times time. We might have a good concentration of reactive species at the surface, but how long is the water staying at the surface? It's a legit challenge. So we almost have to design something that's more like an adsorption column where it's constantly in contact. The water's constantly right next to the surface, and our contaminants can attach to the surface there and adsorb. But how do you do that and get light through the whole thing at the same time? It's tricky. Right? So there's several challenges here. Another challenge for the reactive species to find the target is quenching reactions. Hydroxyl radicals like to react with just about anything. So this could be natural organic matter, or it could be even bicarbonate, HCO3 minus, which is pretty much in all of our natural water, right? This is the CO2 dissolved at near neutral pH, but we get bicarbonate. And that tends to react pretty strongly with hydroxyl radicals. So it's going to be something competing with our targets for the chlorine, so to speak, right? So all of that's kind of tying into achieving that necessary contact time. Now, in addition to all this, we also have to find a way to keep our catalyst there, right? So maybe we're coating it onto something. We have to double check that it stays on the coating so we're not drinking the catalyst or the photosensitizer, this fullerene nanomaterial that might be super toxic, you know. I think it's not so much, but you know, there are legitimate concerns and we need to worry about that, right? So we have to have a fancy container that does contain our catalyst and also lets water flow through nicely and light reach everywhere, right? So this fancy container has to do a lot of fancy stuff, right? That's kind of the key. Okay, just a note on the diffusion lengths we might expect and how reactive the different things are. So here's a few reactive species we could identify. Hydroxyl radicals having a really high redox potential, so they're quite reactive. The superoxide radical anion, also quite reactive, not very stable. Single oxygen also has pretty good, pretty good reactivity, but has a bit further diffusion length. This has to do with how long they last in water. So we also sometimes will produce hydrogen peroxide. That lasts a good long while. So that's probably can diffuse. I don't know if this is accurate or just a sketch here, but it's reasonably reactive, and it can go pretty far. Some advanced oxidation processes actually just use what we call Fenton reactions, which would be H2O2 plus iron, and there's some similar ones that can do it, metals. And essentially we end up with hydroxyl radicals. And so we can create, without any surface dynamics, we just create out in the bulk solution a bunch of hydroxyl radicals if we do those together. And surprise, surprise, this is what you've done if you've ever poured peroxide over a scrape. And then it fizzes. You're getting a strong reaction because there's iron in the blood. If you poured peroxide on your skin with no blood, you're not getting any fizz, right? Now, if you happen to, like, I don't know, put some iron there of some sort, maybe some rusts, maybe you will get fizz. I haven't tried this, but I bet you could get that to fizz as if you have a cut. But you pour it on a cut to try to disinfect it, and it's working really well because peroxide's pretty reactive, but especially because now suddenly you have a whole bunch of highly, highly reactive hydroxyl radicals. Now, you wouldn't want to do this forever, for a long time, because then you're just going to burn out your flesh there, just degrade it with all these hydroxyl radicals, right? So you just want to do it a bit, kill off the microbes, don't let any infection happen. But that's what's happening, is you're doing exactly this reaction. Okay, so to highlight a bit this quenching effect and the role of dissolved organics, here's a bit of work from a recent publication we had last year, where we took, this is TiO2, so we have a surface, photocatalytic surface, and we had this molecule coming and trying to be degraded at or near the surface, adsorbing onto the surface, but we also had organic matter that might interfere. And what we're seeing here is an illustration of, okay, we have a few different organic matter types, so humic acid, some little colloidal material, some couple other fractions we took from wastewater and said, okay, if it sticks onto this thing, it's kind of hydrophobic. If we stick it on this thing, it's really hydrophobic. How are these interacting with our surface? And so we have different stuff happening at different pHs. And so we see sometimes the process is actually improved. If it's going up from the starting points, the stuff is actually degraded better in that situation. So in some way, the organic matter helped take the molecule and bring it to the surface. It just created a more favorable interaction for it, or environment for it to come to the surface. Other times, most of the times, it drops it because of this interference. So we're really investigating, and we can go into all those details, but essentially, sometimes that organic matter is a big problem. In other conditions, maybe the charges are set up correctly. Maybe at certain pH, this is going to be positively charged, negatively charged. The surface is going to be different. The organic matter has its own interaction. So it gets pretty complicated, but this is the way we were studying it to see, okay, how does the amount of dissolved organic matter, that's the x-axis, the amount of dissolved organic matter, how is that impacting our stuff here? It turns out this is very much adsorption isotherms plus quenching competition. So there's like two things happening: competition for the surface and competition for reacting with the radicals. So don't really need you to know too much about all that, but the concept here is we're trying to figure out how can we overcome the problem of doing this stuff in real life, like actually dirty water. We have to be able to do this in dirty water because that's what we want to clean, right? So there's those questions. So another topic would be, how do we keep the catalyst where we want it in the water without it escaping? So it's got to be, we have to kind of treat the photocatalytic material itself as a possible contaminant you don't want to drink. So ideally, we'd covalently attach it, stick it onto a surface, and keep it there, and be able to reuse it. Also quite important, all while achieving some contact time. So one simple way we might be able to do that would be to have a slurry, design it so that it's not too deep so the light can actually reach everything. You could get some treatment that way, and then add a membrane at the end to keep all the particles inside. Now that could work, but if you're going to filter it, why are you also bothering to do all membrane, you did all the photocatalysis stuff, and maybe your catalysts are large enough and it makes sense, it's cheap enough to do that. So the idea here, we'd have the contaminants, add the natural organics, and yeah, so that's the real system. How well is that working? Maybe it requires a lot of time. A possible upgrade to that would be to have water flowing into a tubular membrane that's flowing in an inside-out regime. Water's coming in, sunlight's or UV light or something is coming to the outside of this, and we load the outside of the membrane with a catalyst so that right where all the water has to pass through, just that little area, the water, like all of the water that gets through is going to pass through that area. We energize that and load that with catalyst so that there's high concentration of our reactive species right there. The membrane itself maybe can block some of the organics too. So as, let's say, viruses or pharmaceuticals are coming out, they get blasted right there where all the water does have to pass. So that's one possible concept project that I helped with a bit during my postdoc, and it kind of worked reasonably well for that. And here's some data on that. We did see a bit of an improvement for human adenovirus destruction, but excuse me. But it wasn't super impressive, all things told. The membrane itself was actually removing some of the viruses, so I mean, perhaps that's already helpful and part of why it was not as impressive. So it was definitely a neat thing. So another concept would be to, and I kind of mentioned this, maybe pack a transparent column that we can shine light through with transparent supporting bead materials. Or possibly something like this, and a colleague in the chemical engineering department, Dr. Kevin McPeak, has been working on this. Maybe take some sort of a fibrous material made out of quartz so that it's transparent, and transparent to the UV even. So if we could do something like that and load our system with our catalyst, we can kind of do this combination of adsorption and photocatalysis at the same time. It would almost be like having an adsorption column that's self-cleaning, which could be pretty nice. So there's some promise there. If you can do all of that, you get your light management decent so that your light can actually get to all the catalysts. You can have good surface area, good contact, and you could have, assuming you can get your catalyst to stay there also, to really embed it there and keep it there. So that type of solution seems to me like the way forward to something practical. So I'm hopeful that it could maybe get there. Some other kind of opinion articles over recent years, certain factors that people have discussed thinking are important. It's a funny photo here of my PhD advisor that he drew of himself, cartoon. He's a really fun guy, and is an amateur artist in addition to being a prolific researcher. Another possible concept, I've seen a little bit of work here, might try to do a little more on this myself, is to use some sort of a fiber optic cable. And then, so fiber optics are designed to bounce the light back and forth so that they can transmit and keep the photons in the cable itself. Well, maybe you can remove some of the cladding or the casing that prevents the light from leaving and add catalyst there instead, so the light is kind of bouncing along. Some of it gets used up by the catalysts, but otherwise is continuing to internally reflect. And then you just make a lot of that, high surface area of that, and your photocatalyst system, you could directly get your light transfer exactly to where you need it without any waste, and potentially design high surface area from there. So some neat concepts out there, neat possibilities. A couple other things I've seen. Sometimes the photosensitizers could be used for a specific chemical reaction to as a way to synthesize some specific chemical. This would really be that single oxygen route. A few chemicals can be synthesized this way, so maybe you'd like have a channelized system like that, some concept. Probably not as useful for water treatment, but maybe some concept like that, perhaps. Okay, any questions? So it's definitely something that I've done a lot of research on, been excited about, but it's also, you know, still you can't go buy a product that does photocatalysis, right? This is designed for that. So hopefully we'll get there. Certainly has been interesting, and I think lot of neat progress, a little bit of hope that it could become practical one day. All right, thank you guys. We'll see you on Thursday.